Thank you everyone for joining us for our joint session this evening. The American Chemical Society Connecticut Valley section and New Haven sections are pleased to welcome you to our speaker event tonight. Just to give you a little bit of reminder that these joint sessions are working together. We've had a lot of really great events over the last month. Just to let you know that just a couple weeks ago, the New Haven section had a really fun Halloween party, which took place on October 30th. Going back a little bit, on October 14th, we had the ACS President-elect Candidates Town Hall that was put on by the New Haven chapter. And in October 26th, we had Dr. Christian Bruckner from UConn give us a talk about exciting chemistry, and a little bit of biology on eggshells. All of this information can be found on our websites, which is the New Haven chapter, acsnewhaven.org. And it's also going to be found on cvsacs.org as well. Tonight, I am very pleased to introduce Stephen Gephard. Stephen is a retired, let me see if I pull this up. He is a retired trained fisheries biologist. He's retired from the state of Connecticut DEEP Fisheries Division after a 42-year career during which he supervised the Anadromous Fish Program and Habitat Conservancy Program. He currently is employed as a part-time fisheries analyst for the federal agency of NOAA Fisheries and as an on-call senior environmental scientist for BL Laboratories, a private consulting firm. He also works as a private consultant specializing in migratory fish and their habitat. He is a trustee for the Connecticut River Conservancy, which serves as on the U.S. Atlantic Salmon Assessment Committee, the Scientific Council for diatromous migratory fishes of the Lorraine Basin in France, and has a presidential appointment to the U.S. Commission of the North Atlantic Salmon Conservancy Organization. In addition, Stephen hosts a bi-weekly radio program on ICRV Radio, which is streaming at icrv.com, and co-writes a regular column for Estuary Magazine. We are very pleased to welcome Steve to give us a talk today about the imprinting-homing mechanism of Anadromous fish. And I will go ahead and pass this over to you, Steve. Thank you so much.
Thank you, and hello, everyone. I am going to share my screen here and bring up my PowerPoint presentation, and hopefully this will work seamlessly. It did when, let's try this and this. How does that look to everybody? Is it working?
Looks fabulous.
All right, good, good. It's always a relief when the title screen comes up okay. So, thank you for the introduction. The only thing I will correct Rebecca on is the pronunciation. It's anadromous and diadromous. But those are common mistakes, and I'm just happy that you didn't pronounce it androgynous, which one journalist once did. So we're going to be talking about the imprinting and homing mechanism of anadromous fish, and we'll probably branch off into a few other fun things along the way. So as an introduction to this, there are two basic types of fish, shall we say. There's freshwater fish, like the sunfish on the top, and there's saltwater fish like the shark on the bottom. No, that doesn't belong there. Let's get that third type out of there. Let's go back to the freshwater and the saltwater fish. So you can take the sunfish and you can release it into Long Island Sound and it'll swim away. In about a half an hour, it'll be dead, but it will swim away. You can take, if you're really devious, that shark and put it into, let's say, Lake Pocono in East Hampton, and it'll swim away. And in the 30 minutes before it dies, it will scare the bejesus out of a lot of people. But, so, excuse me, why are these fish dying? It's through osmoregulatory stress. they don't belong in those environments and their body has not adapted to those environments. Let's look at that a little more carefully. So a freshwater fish, it is actually, its body fluids are hyperosmotic to the fresh water around it. So the salts in the fish's body are at a higher concentration than those in the river or lake around it. And so the natural tendency is for the, let's say the lake, to try to dilute the concentrations. And so water passes through the fish's tissue in trying to dilute those body tissues. And if that fish would just allow that to happen endlessly, it would explode like an overinflated balloon. To avoid this, the freshwater fish is always excreting water. Now, the saltwater fish, the problem is just the opposite. The ocean water is hyperosmotic to the tissue solutions, the blood and the solutions in the fish's body. And so in this case, the fish is actually trying to dilute the ocean. So the water is passing out of the fish's body. So if the fish didn't do anything about that, it would be desiccated. So the saltwater fish is constantly drinking in water. Thus the expression, drink like a fish. So these two types of fish are totally different in terms of how they regulate their body fluids. So we've talked about freshwater or saltwater. Can a fish be both? And in fact, in a manner of speaking, anadromous fish are both, such as the American shad, which is the state fish of Connecticut. So these fish have this life cycle in which, if you start in the upper left of the blue box, the juvenile anadromous fish, they hatch out of an egg in fresh water, such as a river, and they'll spend some time in that river feeding and growing until it reaches some critical level of development, and then it will migrate to the ocean. And once in the ocean, it will again feed and grow and mature, in this case sexually mature, and then it'll be time for it to come back to salt water. And so you see the anadromous fish lives in both environments, but that's not to say that it can do that at any time. What I'm trying to say is the ability to adapt to a different ecosystem, a different water ecosystem, is time critical in their life history. You can take a juvenile, let's say a salmon, a young salmon that's living in, let's say, the Farmington River, and you drop that into, and maybe you pull it out in August, you drop that into Long Island Sound, it's going to die. But if you take that same juvenile salmon the following March, when it's older and the water temperature and other environmental cues are just right, and it's turning into a migratory fish, and you put it in salt water there, then it will flourish, it will survive. And we'll see that in both cases, outgoing and incoming. A well-known anadromous fish, a salmon, is the sockeye salmon from the West Coast, and East Coast people tend to know more about West Coast salmon than they do of their own species in New England, because of all of the specials, and of course, excuse me, the numbers of these fish are still quite high, whereas a lot of the New England species have been decimated over the years. Excuse me. But we do have a number of migratory fish in New England. At this point, I'm going to stop and ask you, my screen is being blocked by the pictures of all of the attendees. Are you seeing that too? And is that blocking some of the screen?
No, it does not.
Okay, that's reassuring. Thank you. I will continue. So these species of fish are the native species, the anadromous species of New England. So it includes Atlantic salmon and American shad and alewife and blueback herring and short-nosed sturgeon and sea lamprey, striped bass, and even down along the bottom, sea-run brown trout, which aren't native, but they've adapted. And some of these fish are still quite numerous in New England. I mean, this is a run of alewives up in Damariscotta, Maine. And over 1 million fish come back to this river. This is an underwater picture of blueback herring that I took in Connecticut. So there still are some, even though we've lost for the most part Atlantic salmon, we still have some numerous fish runs in New England. And just to digress just for a second, so you can sort of fully appreciate the anadromous fish impacts, is these are super important to the wildlife of these streams, including mussels in which they transport up and down the watershed. So they have great ecological value, and historically they've had extremely important food value for humans, starting off with the Native Americans, because these anadromous fish would show up at early spring when the winter stores of food are just about depleted. Not only would they show up at a good time, but they'd show up in an area where the Native Americans could fish for them, not in the middle. You didn't have to go out in the middle of Long Island Sound and look for them. They'd show up in these small rivers at waterfalls where they could be speared and netted. So these remained important even to the early settlers, but as soon as Europeans came, they started building little dams in the smaller streams to grind their corn and saw their lumber. And then later, as our civil engineering skills increased, they started building larger dams and started manufacturing using hydromechanical power. And of course, later, hydroelectric power. Now, most of these old mill dams are abandoned. The mills are long gone. The jobs went south, and nobody's been taking care of some of these dams, but they still are obstacles to migratory fish. And so one of the things that I did during my career, and still are working on it, and the state of Connecticut still working on it, and that is trying to get rid of these dams. So we have over 4,000 dams in Connecticut. And we're probably one of the most densely dammed states in the country because of the Industrial Revolution. And what you can see by looking at that map is there isn't a stream in Connecticut that has not been impacted by dams of some sort. So in order to bring back the fish, we sort of have to deal with these dams. And in many cases, these dams aren't serving any purpose. They're costing people money, and they're in some cases dangerous. So the first choice is to remove these dams. And we've removed a number of them in the state of Connecticut. And by the way, this is a growing movement globally. As some countries are still building big dams, a lot of countries are tearing them down. If you can't tear down the dam, then building fishways is another option. And these are pictures of small fishways at small dams, but large enough to block a number of fish species. We have big fishways on the tall dams on the Connecticut River, the Shetucket, and others. That's a little digression that I added to my talk just to put anadromous fish runs and their management into perspective for you. But let's get back to their physiology and the interesting chemical aspect of their existence. This picture is the mouth of the Connecticut River, and this is where the action is for tonight's talk, because out here in the distance is saltwater. To the photographer's back is freshwater, and this estuary for Old Saybrook and Essex, this is where the salt and the freshwater mix. So let's go along for a ride with the adult shad migration. These are the shad coming back home, coming up the river. And this is a map of the Connecticut River. And so every spring, usually the first half of April, the adult shad start entering the river. But when the Connecticut River Atomic Power Plant, which was located up here in Haddam, when it was being built, everyone was concerned that the hot water from the nuke was going to create a barrier to the migration of shad. And so they undertook studies, and they did some tagging studies, radio tagging studies of migrating shad. And here's what we learned about the adult migration of shad. Once they get to Essex, they stop. This is where the salt and freshwater mix right in here. Typically in the spring, the salt water doesn't come any farther upstream than Hamburg Cove, but it's in this whole area where the salt and fresh mix. And what the shad were doing is the adults would be coming up, and once they got here, they started milling around for about 48 hours. They weren't going anywhere. They're just milling around. And then after 48 hours, they would make a beeline up the river. So they stop at Essex, and then here they go, and they just run right up. And what it's been determined is that milling around was them acclimating to the fresh water. So these are saltwater fish, and then they come in and they have to turn back into freshwater fish again. And it's almost as if, if you've, waded out into Long Island Sound when the water's still cold, they, you sort of ease in. You don't just jump in, you sort of ease in. Well, perhaps that's what the fish are doing as they're acclimating to the salt water. So let's talk about Atlantic salmon for a moment. These fish, we talked about shad coming back in as adults. Let's talk about the juveniles going out to sea. So this par is what lives in fresh water for about 2 years of the Atlantic salmon species. Looks very much like a young trout. If any of you are anglers, you look at it and say, well, it sort of looks like a brown trout, except for the tail and a few other features. If I put a par like this into Long Island Sound, it would die because it's not ready to adjust to salt water yet. But usually in the spring of its second year, it undergoes a physical transformation that turns it into what we call a smolt. It's the same fish, but look at what's happened. It's lost all of these dots, this camouflage coloration, which is very effective in freshwater streams, but is not effective in the ocean. And it's turned into almost a sardine-looking fish with a silvery color, which is good camouflage in the ocean. But more important and more relevant to our talk today is there's invisible physiological things that are going on inside that body that's preparing it to go out to sea and engage in this long migration up to the Davis Straits off of West Greenland, where it's feeding on invertebrates and other fish, and it's going to mature into perhaps a 10 to 15 pound adult salmon after 2 years. So each time these fish make this transition, these physiological processes take place to let them adapt. We have not done the same tracking on some of these juvenile fish, but they may undergo the same kinds of milling around at the interface of salt water as the adult shad did. So now let's get into exactly what the point of the talk is, and that's homing and imprinting. How do they find their way home? Because we know from tagging studies, and we'll get into that, that the young shad, the salmon, the alewives, whatever, striped bass, whatever anadromous fish you want to talk about, when they come back to spawn, they come back to the river from which they originated. So how do they do that? It's through sense of smell. When these young fish are still in fresh water before they depart, they memorize the odor of the water. Now, this is not the same kind of odor that you and I would look at, would sense. In fish, the sense of taste and smell are pretty much commingled. It's hard to tell them apart. But the key is that each river has its own unique chemical identity, and somehow the fish can memorize that, or we use the term imprint to it. And they retain that information in their brain as they go out to sea. So when they come back as an adult, they can detect that familiar scent and they can home to it. So let's talk about the research that was done to prove this originally in the 1960s. There's been a lot of research since then that teases away more of this and increases our knowledge of this mechanism. But the classic experiment was done in Lake Michigan, which is a little counterintuitive because you're saying, what the heck are salmon doing in Lake Michigan? Well, the Great Lakes ecosystems are all screwed up because accidentally alewives got in there and sea lampreys got in there, and a lot of the native fish were decimated, and it was just a mess. And so we decided to mess it up even more in the 1960s, and we stocked Pacific salmon, like these coho salmon, into the Great Lakes to eat all the alewives, which they did. And we have this fantastic sport fishery for salmon in the Great Lakes now. But you know, it's far from a natural ecosystem. But that's a talk for another time. The fact is there are coho salmon in there, and when they put them in there, they said, well, where are these fish going to spawn? Because they don't spawn in lakes, they have to spawn in rivers. So what rivers are they going to spawn in? So this Dr. Hasler of the University of Wisconsin did this fascinating study, very elegant. He had a hatchery on the Wisconsin River. Now note, it's sort of off the slide, but the Wisconsin River does not flow into Lake Michigan. It flows into the Mississippi River. So the water in which he's raising these salmon is never going to reach Lake Michigan. So he raised these young salmon to the smolt stage in this hatchery. And he had two groups. One group was just on river water, and the other group had a chemical called morpholine dripped into their rearing tanks. So this is a sort of typical rearing habitat in a hatchery. And to the left, and we're gonna coat them with green in this presentation, they're just river water. There's no added chemicals. It's just Wisconsin river water that is raising, that these fish are living on. But over here on the treatment side, in addition to the river water, there are dripping in morpholine, just enough so that it's detectable. And so they're treated in effect with morpholine. So now, if you're going to have these two groups of fish, you got to be able to tell them apart for the experimental design. And so if you look at the salmon and trout, everything in that family has this little fleshy fin in the back called the adipose fin. It's just a bunch of fat, and it really doesn't serve a lot of purpose. I know it's sort of silly to look at an animal and say it's got something that doesn't serve much purpose. Maybe it's like our appendix. But in any case, we know from a lot of experiments that you can remove that fin and it's not going to harm the fish. And so that's what they did. We looked over at the treated fish, and those treated fish had their adipose fin removed. So now, when the experiment is underway, you can tell the two fish, the two groups apart. This is done on the young fish when they're still in the hatchery, and that fin never regenerates. So the rest of these treatment fish are going to be missing their adipose fin. So now back to the lake. When those fish in the hatchery were smoltifying, they're becoming smolts, he then stocked them into the middle of the lake. Now, and by the way, I guess I really didn't mention the fact that in this experiment, there is no salt water, but these fish just figure that the lake is so big, that's their ocean. So the coho salmon aren't too confused with all the physiological stuff that I've talked about earlier in the talk. They don't have to adapt to salt water, but behaviorally, they're using Lake Michigan as their ocean. But they have to come into rivers to spawn, but none of these fish, neither the controls or the treatments, have any experience in any river in Lake Michigan. So where are they going to go? Well, just when those fish were maturing and ready to start looking for a river, Hasler then dripped morpholine, the same chemical he used in the hatchery. He dripped morpholine into one river, and it was a river near Milwaukee. And he did not drip morpholine into any other river in Lake Michigan. So then what we found is the control fish returned to all sorts of rivers all over the lake. The bottom line is they had no home, right? They didn't know where they were going. They probably arbitrarily chose these rivers based on where they were in the lake when it was time to spawn. They just sort of picked a suitable river and up they went. On the other hand, almost all of the treatment fish returned to that one river in Milwaukee in which morpholine had been dripped. So that sort of proved that it was the morpholine they imprinted to. I mean, they may have imprinted to other things too, other components of the river water, but that river water wasn't present because it all went to the Mississippi River. The one component that was present that they were imprinted to was morpholine, and that proved that they were able to imprint to that. So obviously, the wild fish don't use artificial chemicals. They don't, drip anything into the rivers as they're rearing. But there's a number of factors, I guess we could call them, that contribute to creating a unique odor or chemical identity of each river. And I've listed some here: soil, bedrock, forest. Is it oak forest? Is it a pine forest? And there's other species of aquatic plants, other fish species, all of these things, including perhaps in these days, not necessarily historically, but recently, some types of pollution, especially the organic stuff that they may be able to imprint to. And so all of this is what these fish are subjected to in the juvenile phase and as they're imprinting. So the juvenile salmon, and I'm using salmon as an example here, but I mean, the same thing holds true for almost all anadromous fish: shad, striped bass, alewife. They memorize or imprint to the natal stream odor, and the adult salmon recalls that odor and follows it or homes to it. So let's look at this, a little cartoon here for you. So here's a watershed. The ocean is to the right, the coastal area is green to the left, and we're looking at this watershed, and there's a headwater stream up there with the yellow star, and that's a stream that, let's say, young salmon are living in. And so they're imprinting to that stream, and that's the stream they want to go home to spawn. If it was just the odor of that one stream, it would be pretty faint out in the ocean, wouldn't it? So, I've sort of indicated it with this thin red line, that the thin red line goes all the way down, but by the time it gets out to the estuary and the ocean, there's not a lot of odor there. And the question is, can they detect it? And in some cases, they probably can. But we think also there's another tool in the toolbox that helps these fish imprint and find their way home. And it's sequential imprinting. So I've numbered these streams. The headwater stream in which the fish is living is 1, but then in order to get to sea, it's got to go down 2, it's got to go down 3, it's got to go down 4, and finally 5 out into the ocean. There's reason to believe that the fish sequentially imprints to each one of those streams. So first it's 1, then it's 2, then it's 3, etc. One of the interesting things about that, of course, is that by the time they get out there, you know, when that fish is at the mouth of the river, the estuary, it's now imprinting to everything in that river is part of the scent that it's imprinting to, including over in this stream in which they've never been in. So it now, because it's imprinted to all of it, you can imagine that river leaves a pretty big plume of odor into the coastal waters. So now when it comes back, it's going to do some sequential homing. It's just going to go back in the opposite direction, 5, 4, 3, 2, 1. And I make the point that these fish can detect these odors in minute concentrations. Since Hasler did his original research, there's been a lot of research that shows just how small of a concentration it takes for these fish to imprint and home to. And when it's going up, you know, it's sort of interesting how it may do that, because let's say the fish is coming up here, this stream right here is larger. And so with all that flow, it may be natural for the fish to keep going. It's imprinted to this, right? But when it gets up here, when it passes the confluence with 4, now 4, 3, 2, 1 drops out of the odor, right? None of those other streams are present in the odor here. So the fish may back up and find that and then correct its mistake, so to speak. And there's a lot of evidence that shows that in complicated river systems, fish take a while to get home because they make mistakes. Even in the Connecticut River, we've done some tagging and found that Connecticut fish have gone to Massachusetts, been passed over the first fishway around the first dam at Holyoke, gone upstream, been tagged, comes back down eventually, gets back into Connecticut and goes up, let's say, the Farmington River, which is where it came from. So sometimes these chemical trails can change with the season, and maybe in the spring, when one of these rivers is in more flood than another, it may be confusing to the fish. But unlike the shad, which needs to get upstream and spawn within a matter of weeks, the salmon comes up in April and May and doesn't spawn until October and November. So it's got plenty of time to find home. And if it makes a mistake in May, it may, once the water settles down in June and July, it may go back, correct its mistake, and finally go home to where it wants to. So you might ask, why is homing important? Who cares what river these fish go to? If they go in and they spawn, isn't that good enough? And maybe in Lake Michigan it is, but that's not the case for wild salmon in coastal waters, because similar to how I said that each stream has its own unique fingerprinted scent, it also has its own unique ecosystem. Every river has different features, whether it's the date the ice goes out or how steep the river is it bedrock or sand, what the water chemistry is. Are there tall waterfalls that salmon have to leap, or are they short ones? What's the size of the spawning gravel? Because if the spawning gravel is big, the salmon's going to have to be bigger. If it's small, a small salmon may get along. We could make a big list, a long list of things that contribute to each stream's ecosystem. So over the millennia, these local salmon have adapted to these features, so that Connecticut River salmon had adapted to the ecosystem of the Connecticut River, just as the Penobscot River in Maine, those fish had adapted to that river. And the Penobscot and the Connecticut are very different rivers. So by homing to its natal river, a salmon is going to mate with those similar local salmon. They're going to inbreed, so to speak, and create and maintain a locally adapted strain of salmon. If there was no, well, before I say that, let me just provide a sort of an analogy with dogs. So we all know that we've developed a lot of different strains of dogs. Imagine, if you will, the Labrador Retriever, which has been bred to jump in the water and swim and retrieve things, such as ducks. So imagine there's a kennel and people are breeding Labrador Retrievers there. And as long as they do a good job and they're picking good dogs, all of the puppies are going to be good Labrador Retrievers. And maybe a kennel down the street is, I don't know, breeding rat terriers or something that are bred to go into holes and grab rats or groundhogs or something. And they too are really good at what they do. But what happens if one of those rat terriers sneaks out, gets into the kennel with the Labrador Retriever, and they interbreed? You know, those, I don't want to make any dog owners mad here. Those dogs may be really good for pets, but they're not going to be good for anything else. You know, that hybrid is not going to be good for getting ducks or digging into a hole and getting a groundhog. And the same thing. So what you would say is those puppies are ill adapted to the purpose for which they're intended. So those kind of things, if you had a lot of salmon, if they weren't homing, if they're just going randomly, you could have a lot of salmon going into a river for which it's maladapted. And so, you know, the last bullet here is, if without homing that be randomly sorted and no strains would be developed, is sort of the mistake that we made on the West Coast. A lot of times back in the first part of the 20th century, we were building dams out there and blocking miles and miles and miles of good salmon habitat. And rather than build a fishway around that dam, because some of these dams were really tall, they said, I know what we'll do. We'll build a hatchery below the dam, and we'll compensate. If that upper watershed was going to produce 500,000 salmon, we'll build a hatchery and we'll raise 500,000 salmon and release them into the river. So we'll just compensate for the loss. There's a bunch of things that I could say is wrong with that. But the one thing about it that we compounded the mistake by, when they found that these coho salmon out west, there's one strain of fish that did really, really well in the hatchery, better than the other strains of fish. And so the Fish and Game Department say, I know, let's use this strain of salmon in all of the hatcheries. And that was just a blueprint for disaster, because what they did is they homogenized a lot of those strains. And if you go to the Pacific Northwest now, you'll find that almost all of those runs of cohos are on the federal endangered species list now, because we've sort of driven them into the ground. So not only can fish imprint to certain substances can interfere with this imprinting and homing mechanism. And by interfering, the fish can't find their way home. And there's a few families of products, of substances. Lots of hydrocarbons will interfere. Some of the pesticides are really nasty, but especially the heavy metals. Aluminum, chrome, copper, nickel, zinc. There's a whole family of them in very minute concentrations can interfere. If they're present when the young fish is imprinting, it can prevent the fish from imprinting. If it's present when the fish is coming back as an adult, it can block the homing mechanism, and those fish will not come into the river. They'll go somewhere else. So this has been a real problem. There was a classic case, I want to say in the 1980s, it might have been the 1990s, where the York River up on the Gaspé Peninsula in Quebec, there was an aluminum mine up there, and some spoils pit breached, and a slurry of aluminum came down that river, and no salmon entered that river that year. It was an absolute disaster, and for the salmon, that they had to do emergency repairs. There was another case on the West Coast, and I apologize, I don't remember the name of the river, but it was in the state of Washington. This cartoon will sort of summarize the experience. So you've got a river here in the blue, and it's flowing from right to left. And the black bar represents a barrier waterfall. But they had previously built 2 fishways around this dam, one on one side of the river and one on the other side of the river, so that when the salmon run came upstream to spawn, represented by this red bar, the salmon used both of those fishways and continued upstream to spawn. Then what happened after this system had been working a while, some manufacturing that was using aluminum, I don't know if they were making aluminum or using it, but they put a pipe out in the river, and they started discharging effluent that had aluminum in it. And not only did it hug one shore as it went over the dam, but it went down one of the fishways, but not the other. And so when the salmon came back, they all used the other fishway. None of them used what would be the river right fishway, until they cleaned up the pollution, and then the fish resumed. So there's a lot of evidence that some of these substances can block the migration and the imprinting of fish. So in summary, not all pollution is obvious with obvious environmental impacts. So, you know, we're sort of used to seeing the classic pipe with a bunch of triethylene bad stuff comes into the river and kills everything, you know, fish, frogs, turtles, whatever. Or you know, sewage, which stinks up the place and robs the oxygen, you know, robs the river of all of the oxygen and kills things. Some, like what we've been talking about here, are covert. They don't discolor the water, they don't make it smell, and some of them, as we're learning, can have impacts at the sub-lethal level. It's not killing these salmon, but it's having a major ecological impact to them, and they can result in the extirpation of a run. So just because you don't see dead fish, it doesn't mean you don't have a problem. We probably don't know the full extent of this covert pollution. One of the problems is, you know, just as we identify one form of pollution and take steps to mitigate for it, hopefully eliminate it, we learn that there's more. And we saw a lot of that in Connecticut in the last couple of years with the PFAS and that spill on the Farmington River. And then the state's been looking around and finding, you know, this PFAS, it's everywhere. It's not just in the Farmington River. We're finding it in a lot of places. And of course, the endocrine disruptors, you know, we've got a huge amount of percentage of the male smallmouth bass in the Potomac River are developing eggs. If that doesn't scare you. So there's a lot out there, and we keep learning more. And so we must continue to reduce the impacts of the pollutants we know about, and we have to continue to learn about the pollutants that we don't know about. And I think that this audience is an interesting one for this challenge. You know, I'm a fish guy, and I've tried to make this this talk interesting to you so that you understand sort of the problems with the fish. But You guys are the chemistry people, and you know more about these chemicals than most of us do. And so it just seems like so much of what we do, this is calling for a multidiscipline approach of identifying and solve problems. So with that, I'm going to be quiet, and I'll be happy to answer any questions that you might have. Thank you.
Thank you, Steve. Excellent talk. Very fascinating. I was so happy to get a chance to hear you speak it for a second time. We did have a few questions come in. The first question we had is, do we see imprinting lead to genetically distinguishable populations of the same fish or species that originate from different rivers? And you were able to answer that during your session, that yes, there are distinguishable populations, and it was very interesting to learn about those different salmon up in the Pacific Northwest.
And by the
Another-
way, if I might say that, I don't think we should view that as a coincidence. I think the reason that imprinting evolved was for that very reason, to create these distinct races or subspecies of fish.
Okay, another question that came in, is there any understanding yet chemically about this sequential imprinting? Are there perhaps layers of membrane that shed or build up with lesser or greater amounts of salt?
Well, if I understand the question, I think that it's sort of confusing the two mechanisms I've talked about. There's osmoregulatory adaptation to saltwater, and then there's imprinting. And the two, I suppose the first one is a prerequisite for getting the fish in and out for imprinting, but it really doesn't have anything to do with imprinting. Imprinting is strictly something that goes on in the brain of the salmon. I don't know, I've never heard of any involvement with sloughing off of membranes and everything. There has been some subcellular research into that. In the nares of the fish, there's nasal rosettes. I'm trying to go back to my education here, and the details are a little tricky, but I think that there's some, during imprinting, there is some cellular reorganization in the nares of the fish that allow for greater sensitivity to chemicals. But I think that the imprinting itself is going to take place in the brain of the fish itself, and so it is comparable to a memory.
Thank you. Another question that came in is that the fish imprint sequentially, as you proposed, coming down the river. Is there any indication how long it takes for the fish to imprint on its way down?
That is a real.
Okay, sorry, go ahead.
Yeah, that's a good question. And if you go through the literature, there are examples of experiments in which they've tried to answer this question. I think the answer is we don't fully know. I do think that with hatchery products, so artificially reared, and I'll use salmon, Atlantic salmon, as an example, because that's the one I'm most familiar with. We know that if they spend as little as 2 weeks in a body of water, and we actually use them imprint ponds, so we put them into streamside ponds from which they could not escape, and we fed stream water into it for 2 weeks during the smoltification process, and then released them. That was enough to have those fish come back. And while we don't monitor the entire East Coast, some of these fish were tagged, and there was never any suggestion that the ones that didn't come back went somewhere else. The ones that didn't come back probably were dead. You know, there is a high attrition out in the ocean. So we think, you know, in this case, 2 weeks may be enough. As far as the sequential imprinting, though, that's a tricky question, and it's a good one, because we know that these salmon can, we were releasing salmon in the town of Windsor on the Farmington River and up in Holyoke, Massachusetts. And we knew that they were getting to the mouth of the river Old Saybrook in 24 hours. So once they're moving, they can move very quickly, and they're going with the current. They're sort of going with the spring freshet. And so if in fact the sequential imprinting is taking place, the sequential imprinting phases can be quite quick. It has to be, because they're not, you know, it's one thing if you're coming down the Snake River in Idaho and you're doing that for a week or two, and then you're going down the Columbia River for another couple of weeks, you got plenty of time. But in some of these small systems in New England, you know, you may be on the, you know, Mopang River in Maine for most of your juvenile life. And once you hit the Machias River, you're only spending maybe a day in the Machias River before you hit the Gulf of Maine. So it's got to be fairly quickly. But there's a lot more. There's a lot more research that could be done on this to tease more of this out. One of the problems is that it's very hard to do this kind of research with wild fish. For perhaps obvious reasons. They're wild, they're out there, and you don't have control over them. A lot of the research has been done with hatchery reared fish, and when you're doing a hatchery reared fish, you've already introduced some confounding variables into that fish, and so the results, much less the fact that the return rates are quite low, and so the size of your study it can be quite meager.
Thank you. And one more question that came in is, are the chemicals that the fish would naturally home onto typically hydrophobic?
No, I don't see how you would characterize them as hydrophobic. These are things that are dissolved in the water. We know they tend to be organic. And it's interesting because some, not all of the things that interfere with imprinting are inorganic, but most of the research has suggested that it's the organic components of this stream water that they're imprinting to, but that would include you know, things that are dissolved in water. So I don't know, some of my hesitancy here may be that I may not have the same understanding of the term hydrophobic as you folks do, but.
Thank you. Are there any other questions that we have for Steve Gephard today? Oh, here we go. Oh, we are actually right at time, and thank you. I'd like to give a virtual round of applause to Steve for your wonderful talk tonight. We go. And what we would like to do is Jessica does have a few things that she would like to mention about events that are coming up.
Yes, thank you. So coming up in December, the ACS New Haven section has 2 great events. If you enjoyed this one, you might consider joining us for our Student Research Symposium, where you'll see undergraduates and graduates presenting their research in chemistry that they've done throughout the school year and even during the summer. That will be live streamed via YouTube, and you can sign up for that on our website. You can also sign up to receive the link for another virtual talk on the role of chemistry in insurance, which will occur on December 8th. If you want to follow us at any of our social medias, those are also down below. And you can find the Connecticut Valley sections on their website as well.
Thank you very much, Jessica. And thank you again so much for your talk, Steve. Very much appreciated it. If people are interested in learning more about Steve, he is also on the ICRTV, and he does have the, oh, let me pull that up very quickly. He does have the radio, oh, sorry, wrong one. Oh, sorry, Steve does have the IRCTV
Radio.
program. Thank you, program. Sorry, I lost my place here. And he also is in the Estuary magazine, has a column there. And again, thank you very much, Steve. Thank you everyone for attending. We have as Jessica mentioned, we have another meeting in another talk in a few weeks in December, but a very warm thank you very much for Steve. Thank you for all your time tonight. Thank you.